Differentially expressed genes are important in bulk RNA-seq analysis. With BxGenomics Bulk RNA View, we can do differentially expressed genes analysis easily. In this tutorial, we will use the bulk RNA-seq in the paper p53-p21 pathway activation contributes to the ependymal fate decision downstream of GSEA as an example. In this paper, the effect of GSEA and Macedas function in neural progenitor cells was evaluated. The GSE number for the dataset is 184397, which can be found in the STAR methods section. Open bulk RNA view. Use the GSE number, we can find the dataset in the database. Click on the quick view. The default plot shown in bulk RNA view is the PCA plot of the bulk RNA-seq dataset. Each point represents a sample projected onto the plane, defined by the first two principal components, capturing the largest sources of variation in the data. Samples from the same condition are grouped by color. It helps to assess sample similarity, biological differences between conditions, and potential outliers or batch effects. In this chart, samples within each group are closer compared to samples between groups, and there is no outlier sample. We can start differentially expressed genes analysis. Click on Run Analysis to evaluate the effect of GSEA and Macedas function in neural progenitor cells. We are going to define two analyses. Test 1 is for GSEA versus control. The method DCQ2 is selected. Test 2 is for Macedas versus control. Click on Start Analysis. The analysis will run in the background. After a couple of minutes, go back to the dashboard and find the project of GSE 184397. Click on the project and then click on the analysis. You can find the analysis you made for this project. Click on View Results. When you go to the analysis results, the default figure is the PCA plot. Click on the DEG tab. Click on Test 1. The volcano plot will show up. You can adjust some parameters to optimize the plot. Here I change the max value of y-axis to 60. There are other tabs you can try to explore more about the test. You can also click on the Save to Output tab to save the volcano plot. After you save it, you can find it in the output. You can download the figure. In this example, there is no outlier samples or regrouping needs. If we want to exclude some outlier sample or regroup the samples, we can edit the sample information in the new DEG analysis step. Following this video, we will develop several additional introductory videos demonstrating sample regrouping, outlier removal, heat map generation, and GSEA analysis. This concludes the introduction of differentially expressed genes analysis in bulk RNA view. If you have any question, please let us know.